Good morning student, welcome to our new class. We are studying gametogenesis in human. First of all, we started from spermatogenesis. Spermatogenesis is divided into two parts: formation of spermatid and then spermiogenesis. So today we will discuss about spermiogenesis, means differentiation of spermatid. Differentiation of spermatid. Although the chromosome number in spermatid becomes haploid, but they don't have the functional capacity of male gamete. Differentiation take place in them to convert into in sperm. This process is known as spermiogenesis or spermatogenesis. The change by which the spermatid are differentiated into spermatozoa are much fundamental in the differentiation. Nucleus of spermatid is contracted by removal of water from it. Nuclear shape and all chromosomes situated in it become together and arrange it completely small space. It is necessary to reduce the water of motile sperm. Not only this, unnecessary substances are removed from nucleus, that is RNA, etc. Only DNA or as genetic material remains in nucleus. Only DNA or genetic material remains in the nucleus. The size of nucleus is also changed in different types of animals, in different types, to become swim easily in water and becomes long and narrow from spherical shape. The shape of head depends upon the shape of its nucleus, which are varying in different animals, just like for example, oval and laterally flat in human and bull, dagger shaped in rats, and frog and spiral shaped in birds. What happened? First of all, you can see diagram on the board. Acrosome of sperm is developed. Differentiation of Golgi body. The Golgi body of spermatid is made up of membranous arranged in the center in many cycles which around many vacuoles in the centers. One or more vacuoles increases in size during differentiation. There are so many Golgi bodies are found around just near the nucleus, and they can differentiated with the vacuole of microdens. Body is visible, that is called acrosomal granule. If one or more than one vacuole and granules are formed, then at last all these are fused together. And make only one granulate vacuole remains. This granulated vacuole attaches the apical part of nucleus. The granule increases in size now and it forms the core of acrosome. The fluid from vacuole flow outside and its membrane covers the entire half of nucleus. Its membranes cover the entire half of proacrosomal granule, and nucleus just like bilobed cap. The double layered cover is called cape of sperm. This is called cape of sperm. The remaining part of Golgi body discharged with excessive cytoplasm of spermatid. Acrosome granules have enzymes which are used in dissolving egg membrane during fertilization. So here one mark question is performed. What is the role of acrosome? Acrosome can dissolve. Acrosome have so many enzymes just like sperm lysine. So that can dissolve the egg membrane during fertilization. Centrosome of spermatid is made up of two cylindrical centriole which are arranged in right angles of each other. A groove is formed in middle place of you can listen carefully what I am speaking. Centrosome of spermatid is made up of two cylindrical centriole which are arranged at the right angles of each other. A groove is formed at a mid posterior. You can see the structure of sperms separately. This is the process of formation of acrosome. First of all, Golgi body and vacuoles are attached together and they can make acrosome granule, and this acrosome granule attached to the nucleus in peripheral stage, and after that it increase its size, or we can say morphologically or metacally and functionally spermatid is differentiated into sperm by sperm metamorphosis. In this process, spherical non-motile haploid spermatid develops from spermatocyte is converted into thread-like motile haploid sperm. Nucleus of spermatid forms head of Golgi body, forms acrosome, mitochondria, and the middle piece distal centrosome forms tails. After spermatogenesis, head of sperm is embedded into Sertoli cell. At last, sperms are released by the process of spermiation from seminiferous tubules. The function of the formation of sperm in animals depend upon the temperature, hormonal, mental status, and hormones secreted by anterior lobe of pituitary gland. That is, we discuss first FSH and LH. FSH means follicle stimulating hormone and LH is luteinizing hormone. So these two hormone, luteinizing hormone stimulates Leydig cells by which androgens are secreted. Androgens stimulate spermatogenesis, which follicle stimulate hormones stimulate Sertoli cells, which with some factors help in the process of spermatogenesis or spermiogenesis. Sperms can remain alive for several years in a tranquility, resting state and cold stage, but their lifespan is, you have to remember, 50 to 60 minutes. After maturation of these sperms, they can live in the epididymis for a long year. Lifespan of sperm is 50 to 60 minutes only in active condition, unavailability of food and high temperature. So here this Golgi body or acrosome granule cover whole the nucleus, and the membrane of vacuoles are covered another membrane on it. So a cape-like structure is performed. A cape-like structure is performed around the nucleus. Centrosome of spermatid is made up of two cylindrical centriole which are arranged in the right angles of each other. A groove is formed at the middle posterior region of nucleus in which one centriole is placed, forming a right angle at the longitudinal axis of sperm. The centriole is called proximal centriole. Means whenever we want to see the structure of sperm, what is the condition? So you have learned two things here. One is spermiogenesis and another is structure of sperm. So first of all, here this is the acrosome and this is the nucleus of sperm, and then this is proximal centriole and this is distal centriole. And this is middle part of sperm and this is tail of sperm. So it is tail and this is middle part of sperm. This is nucleus and this is acrosome. Here the role of distal centriole. The role of distal centriole is making filaments, and the role of proximal centriole is to start cleavage after fertilization. So we can see here the structure and spermiogenesis, both the activities we have to listen. You can listen properly. Centrosome of spermatid is made up of two cylindrical centrioles which are arranged at right angles of each other. A groove is formed at the mid-posterior region of nucleus, in which one centriole is placed, forming a right angle at a longitudinal axis of sperm. The centriole is called proximal centriole. The other or distal centriole take the position below proximal centriole, placed in direction of longitudinal axis of sperm. Distal centriole, which forms filament, or it is basal granule of axial filament. Axial filament is the main part of tail of sperms in simple condition. The tail is formed from only axial filament lined by a thin layer of cytoplasm and plasma lemma membrane. In complex condition, tail include other fibers as in mammal. Maximum protoplasm of spermatid is useless for sperm, and it's removed at the time of formation of acrosome at apical part of nucleus, and cytoplasm of spermatid begin to flow in opposite direction, due to which a thin layer of plasma lemma membrane remains intact with acrosome and nucleus. With its tail, it is being formed from middle part at the time of remaining cytoplasm of spermatid is attached with middle part of sperm. After arranging the mitochondria around the basal part of axial filament, remains cytoplasm with reduce of Golgi body, spermates from sperm. At last, a thin layer of cytoplasm remains around the mitochondria in middle part. The separated cytoplasm is destroyed. Means here some cytoplasm is found, and these axial filament are performed by the distal centriole filament, and tail is made up of only axial filament, and these are the mitochondria which found in the middle part of the sperm. These are the mitochondria which are found in a layer, and that layer is called manchet with its cytoplasm, and rest of cytoplasm are destroyed or removed from sperm. So this is also the structure of sperm, and this structure can be divided into three parts. This is called neck, and this is called head, and this is middle part, and this part is called tail. So today we study about spermiogenesis and the structure of sperm. Thank you for today's class.